Welcome to a deep dive, everyone. We're about to unlock some seriously cool secrets hidden inside molecules. It's like stepping into a molecular detective agency, getting ready to solve mysteries. Imagine needing to figure out what a mystery substance is made of. Could be anything. That weird stuff in the back of your fridge, a stain that just won't budge. That's where today's topic comes in. Mass spectrometry.
It helps us figure out what something is made of at the most basic level.
And get this. It all started with a guy named J.J. Thomson, way back in the early 1900s. He's actually known as the father of mass spectrometry.
Yeah, his work was a game changer. He even won a Nobel Prize in physics for it in 1906.
Wow, Nobel Prize worthy. Okay, this is serious stuff. So how does it actually work? What exactly did he discover?
Well, Thomson discovered that you can separate atoms and molecules based on their mass-to-charge ratio.
Mass to charge ratio.
You got it. It's kind of like imagining a racetrack, where particles are separated not just by their speed, but also by their weight.
Okay, I can kind of picture that.
Exactly. So the lighter, faster particles would zip around the track way faster than the heavier, slower ones. That's essentially what happens inside a mass spectrometer. It separates ions based on this mass-to-charge ratio.
I see. So what are ions again?
Oh, good question. Ions are just atoms or molecules that have an electrical charge, either positive or negative.
Right. This charged particles. Okay, got it. So back to this mass to charge ratio thing.
Right. And get this, that ratio is represented as mz.
MZ, two tiny letters with a massive job, it seems. So that MZ value is like a fingerprint for each ion, right?
Exactly. It's like a unique identifier telling us precisely what that ion is.
So by analyzing these fingerprints, scientists can identify all the different components of a sample.
You got it. Even if we're talking about incredibly tiny amounts.
Wow, talk about a molecular magnifying glass. So we've got these ions racing, leaving their MZ fingerprints everywhere. But it gets even more interesting. What about isotopes? Remember those from chemistry class?
Of course. They might seem like a small detail, but they're actually really important for how mass spectrometry works.
Oh, yeah. In what way?
Well, isotopes are like different versions of the same element. They have the same number of protons, but a different number of neutrons, which means they have different masses.
Okay, so it's like having identical twins, but one twin hit the gym a little harder.
Haha, exactly. And since mass spectrometry separates things by mass, it can tell these subtle differences between isotopes apart.
That's wild. So we've talked about these isotopes in theory, but where do they actually come into play here?
They're essential for something super cool, carbon dating.
Wait, like how we know how old dinosaur bones are?
Exactly. You see, carbon-14, which is a radioactive isotope of carbon, decays over time at a predictable rate. By measuring the ratio of carbon-14 to carbon-12 in a sample, which we can do thanks to mass spectrometry, scientists can figure out its age, like a time machine for objects.
Wow, isotopes are like tiny time capsules. And mass spectrometry lets us read them. Speaking of reading, our sources mentioned a case with mass spectrometry analyzing a pork sample, something about cymbuterol.
Yes, that's a great example of how mass spectrometry is used in food safety.
Really? Tell me more.
They use a technique called LC-MS in that case, which stands for liquid chromatography tandem mass spectrometry.
LCMSMS, that's a mouthful.
Right. It's basically a super-powered method for identifying really tiny amounts of substances, even in complex mixtures like food.
Super powered, huh? So how does that work? Break it down for me.
Imagine a two-step process. First, you have liquid chromatography, which separates different components in the sample. It's kind of like sorting your laundry before washing it.
Okay, get that.
And then, tandem mass spectrometry comes in like a super sleuth, identifying each separated component with incredible accuracy.
So it's like having a whole detective team working together. First, they divide and conquer, then they meticulously analyze each piece of evidence. And in this case, they were looking for this cymbuterol, which I read can be illegally used in livestock.
Exactly. And that's where mass spectrometry really shines. It can detect these substances at super low levels, even if there are only minuscule amounts, which helps make our food supply safer.
That's amazing. It's like having a microscopic eye that can spot even the tiniest traces of anything.
Yeah.
But it's not just for food, right? What else can we use this technology for? So we've talked about food, but this mass spectrometry thing, it's got to be used for way more, right?
Oh, absolutely. I mean, we've only just scratched the surface here.
Okay, we'll dig in deeper. What else is out there?
Well, for one, our sources mentioned portable mass spectrometers.
Oh, wait, hold up. Portable. Like you can take these things on the go.
Exactly. It's not just lab-bound tech anymore. Imagine doing all this analysis out in the field.
Wow, that's wild. So no more sending samples off to a lab and waiting for results. What kind of stuff can we do with that?
The possibilities are pretty much endless. You could identify hazardous materials at, like, a disaster site, right?
Yeah, that would be incredible.
Or even authenticate artifacts in a museum.
Get out of here. No way.
Yeah, and even potentially diagnose medical conditions in real time.
Okay, that's straight out of Star Trek. It's mind-blowing to think how far this technology has come. And where's it headed? What does the future hold for mass spectrometry?
Honestly, it's really exciting. Scientists are always pushing the limits.
Give me an example.
We're talking even smaller, even more powerful devices, capable of detecting even more than we can now.
Okay, so even more sensitive, even more versatile. What about the applications? What could we use this for in the future?
Well, think personalized medicine, environmental monitoring, you name it. If we can detect it, we can analyze it.
So we've gone from figuring out how to separate molecules by their mass to handheld devices that can analyze almost anything, anywhere.
It's been a wild ride, and it's only going to get wilder.
That's for sure. Well, I want to thank you for taking this deep dive with us into the world of mass spectrometry. It's been a journey from the lab to the field and beyond.
It's always a pleasure to talk about this stuff.
To all our listeners, we hope you've enjoyed learning about this incredible technology and its potential. Until next time, keep those questions coming, and we'll see you on our next deep dive.